Hello, in this video we will explore the topics for this week, which are basically understanding the safety rules that we need to follow when we're in the science laboratory, and also understanding the way scientists think, the kind of logical thinking process that they go through that we call the scientific method. We'll start off with having a look at safety rules. So if we think of our lab here in safety, I'd like you to yourself come up with a list of probably 6 really important things that we need to remember for the science lab. Could write your rule as a do something or a don't do something. So have a little think about 6 things that you think apply to the science lab, and then check with this list in a minute. Okay, we're back now. This is what I had in my list. Probably the most important rule of the lot is to listen to instructions. So, fairly obvious, but you're using things that you may have never used before, and the instructions will go through how to use it safely. The other important rule is to not create a situation where you could put yourself in danger or others by running around in the science lab. So no running. There's no need for us to be running. Finish. Oops. Sorry, people. No running. Finish that off. The next rule that we might like to think about with our 6 rules is to make sure that we wear our safety equipment, which most of the time for most experiments will be the safety glasses. Really important they stay on all times. If they go off and you take them off, particularly say at the end of an experiment while you're washing up, that tends to be the time actually during washing up when you're more likely to get something splashed on you than during the actual experiment. So it's really important that they're worn until the teacher says that they don't need to be on anymore. When we're looking at particular chemicals, it's important that we smell cautiously. So we don't go and stick our nose in it. We obviously don't eat or drink anything in the lab. There is a small space at the front of the lab for storing your water bottles, where you can wander to the front of the lab to drink if you need to. Sorry, your water. And finally, our other really important rule is tell the teacher if something is broken. So if you say break a test tube, it doesn't really matter that much. We can cope with it, but we need to know so we can clean up the glassware. We don't want you cleaning up the glassware. We'll do that bit ourselves. So that's pretty much the kinds of things you should be thinking about for safety. You may have in your list additional rules, which is fantastic as well, that you've thought further than the list that I've put here. We'll now go on and have a look at the scientific method, the way scientists have thought about things and how they test something out. Well, the first thing that a scientist needs to do is they basically need to come up with an idea. So they may have, so they start with an idea of how something works or how something is in the world. An idea about the world. They might be looking at how something works. They might be looking at something else. After their idea about the world, they then go and find out, has anybody done it before? So what they're really doing there is they are asking the question, is an experiment already being done? Because if it is, then they can find out what happened in that experiment and they may not need to do their own. So they do some background research. In our case at school, we may deliberately skip this step because we aren't after you necessarily doing an experiment that nobody's done before. We're after you doing an experiment that you've never done before, even though someone else will have already done it when they were at school. Start up with an idea about the world, do background material research. So you actually go and find that out. After that, we come up with a statement of how we think the world works. So make a prediction, and there's a fancy word for prediction that many of you may have already heard before, and we call that fancy word a hypothesis. After our hypothesis, we then go and we need to design how we're going to test it out, an experiment. So we'll just go up and create ourselves a little bit more space so I can put that bit in. So we then go and we actually go and we design our experiment. So we work out what to do, we work out what equipment we will need, and then we actually carry out the experiment. So we actually do the experiment next. Carry out experiment. We go up again. Move. Oops. Try and move the whole thing up, people. Not concentrating here. See if I can make the whole thing go up. Now, it looks like this bit needs to move separately. Alright, we design the experiment, then we carry out the experiment. And finally, we analyse our results that we get from the experiment. So we might try out the experiment, we might take some measurements, and we then go and look at the measurements we've got, what we call our data, or our measurement, or our results. And then we ask the question, was our prediction correct? It's going to be tricky. I'll have to move these up in bits, I think. This bit will have to go up, then this bit go up, and we ask the question, was the prediction correct? It doesn't matter if it's wrong. We should never be unhappy that our prediction is wrong. What matters is that we're going through a logical process to test things. Was the prediction correct? Was the hypothesis correct? And if the answer is no, then we need to change the prediction and repeat the experiment. So if the answer is no, we change our hypothesis and we redesign or recarry out the experiment, and if it is correct, which is a yes, then we are finished. So that, as a kind of diagram or flowchart as we call, is basically the scientific method. An example of the scientific method might be, you might have the idea that the taller you are, the more or the longer you can hold your breath. So if we go back up to, let me go and make this smaller, and we go back up to the top, trying to make it all smaller. It's not actually doing it for me. Let's try and magnify myself out. Here we go. So, let's go back to the beginning and let's look at a particular example here. I might have the idea that taller people can hold their breath for longer. So I might go and find out and do so. I'm basically, if you have a look here, I'm basically over here. That's my idea. I'm starting off with my idea that taller people can hold their breath for longer. I may be correct, I may be wrong. I don't know whether I am or not. So I first will find out, is there something else in the scientific literature? Has someone else done an experiment and written it up that supports it? Because or finds out that what I'm saying is absolute rubbish. And if it is, I mightn't decide to go any further. Though in the case of us at school, as I said, we'll often skip that step out so you get the experience of doing it the first time yourself. Then I'll go and make a prediction. And my prediction needs to be written up, the hypothesis, as a very definite statement. I don't use any words associated with I, so I might say in my prediction, taller people can hold their breath for longer, or taller people will hold their breath for longer. Will hold their breath for longer. Notice how definite the statement is. With a hypothesis, you don't sit on the fence. You don't have, could go one way, could go the other way. You make a definite statement. If you're wrong, it doesn't matter. Then I might design my experiment. So I might actually go and measure people's heights, ask them to take in a breath, hold their breath, and record when they start to take their next breath. So I might use a stopwatch, and every time somebody needs to breathe again, call out their time, or they might have a time sitting in front of them or watching the clock, and they record that themselves. Then we analyse the data. When we're looking at data, it's always good to get a collection of data so that one particular bit of data doesn't change the whole sample. Really important when you're measuring with people. So rather than just ask one person to do it, you might get 10 or 20 or 30 people to do it, so that they then make sure that if there's one person that's slightly different from the average person, that they aren't going to affect all the data, that you're actually going to get a good cross-section of people. So particularly when you're working with people or animals or living things, where every living organism is different, it's really important to take lots of measurements. Then we go and we analyse the measurements, and by analysing the measurements, which I'm thinking over here, we might go then and work out an average or something. We might put the measurements into some kind of table to make them easy to understand and analyse. We might even go on the computer and get it to work out an average for us using a program such as Microsoft Excel, or if you're on your iPads, numbers. And then finally, was the prediction correct? If we do find that the taller people had a bigger number for their breath, which we could find out by drawing an appropriate graph, and then we would be correct. If not, we might have to think about other reasons that makes you hold your breath for longer, nothing to do with your height. So we'd then go back, if we were wrong, and change our hypothesis and have another go at doing the experiment. It may turn out that we don't have to do the whole experiment again. For instance, we know how long each person now will hold their breath for, but if there's something else we want to measure next time, like for instance, instead of their height, possibly the width of their head, then we might have to go then and measure the width of our head if that's our new hypothesis. It doesn't matter whether the hypothesis is correct or wrong. What does matter very much is that we make sure that we have a strong hypothesis and we test it in a fair way. So I'm now going to talk about fair tests. So go into a new slide here and have a look at what it means to have, what it means to have a fair test. So to have a fair test, we want to make sure that we keep everything the same except what we are testing. So for example, if I want to find out if a particular fertilizer works well for growing plants, then everything else about growing the plant I want to keep the same. I want to make sure that I water all the plants the same way. I want to make sure that they get the same amount of sunlight, so that the only thing that is changing is the amount of fertilizer that I'm putting on them. So we come up with some words to explain the idea that I'm going to change one thing. And these are complicated words. So this is all about fair testing. This is all about what's a fair test. And we come up with some complicated words, but they're important words because all scientists use them. So the thing that is being changed we call the independent variable. In our case, thinking about the situation that we had here, our independent variable in our situation would be the amount of fertilizer. So we'll need to record how much fertilizer we're putting on. So that might be our independent variable. And then after the independent variable, let's move down a bit, we then look at the next thing, which is the thing we are measuring. So the thing being measured, or the item being measured, thing being measured, we call a dependent variable. So we'd need to come up with a way of checking how well the plant's growing. One way we could do that is by measuring the height of the plant. Other ways you might like to think of, in fact I'll give you a moment to think of them, other ways we could decide on choosing height of the plant. Give you a little pause where you can think of some way. So have a pause it and have a little think of other ways you could measure how big the plant is. Okay, what are some ways we could come up with and check with your list? A way that you could come up with is you can set a measurement height. You might measure the number of leaves the plant has, or you might actually at the end of the experiment rip the plant out and actually weigh how heavy the plant is once the plant has been properly washed and dried, so that if it's got extra soil on it doesn't add to the weight. You could decide on measuring instead not how tall it goes but how wide the plant grows. So there's many different ways of measuring success, but you in your experiment need to be very explicit about how you want to do it. So you need to say, in this experiment I'm going to do it this way. You won't actually use the word I when you write it up, you'll just say it will be done this way. And often when we have an experiment that involves working with living things or people, we often talk about a new word that we need to be aware of, which is the control. Just move this up again. I have to do it in two spots, I think. And the control is finally the starting off situation. So for instance, if you're testing people, you might decide, for instance, you want to know whether a particular drug is good at, say, for instance, bringing down fever, you may decide that you want to make sure that it is actually that drug that's doing it. So there are some people that you don't give the drug to. So some people take a pill that they think is the drug but isn't actually the drug, and you call those people the control. And the pill that they take, which is a pill that doesn't contain the thing that you are testing, is called a placebo. So they are taking a pretend pill. So they think they're getting the drug, but they're not actually getting the drug. So you know that it is in fact the drug that's doing what you want it to do, like for example bringing down fever. So sometimes with the independent variable, we the sort of starting off situation where there's, say, no fertilizer, when you're not adding anything at all, you'd give it a special word, you'd call it the control. So you'd say that's the control. So often when you're working with animals or people or plants, you will have a situation where you don't have anything there. And that not having anything there we call the control, so that we know that what we are testing by having something there is actually making the difference rather than something else. So this has been quite a long video, so I just want to run through in summary the key ideas. So the key ideas to my mind are very much in what we're doing today as key ideas. The fact that the hypothesis is strong. Hypothesis is a strong prediction. Strong prediction. And you keep everything the same. Except for the change, the one change that you are testing in your experiment. As I said before, if it's testing fertilizers, everything else about the plant's got to use the same plants, the same type of plants, give the same amount of sunlight, the same temperature, the same amount of water, so the only thing that changes is the fertilizer. That's pretty much the summary of our thinking for this week. You've now got an exercise to do.